Around a black smoker, crimson plumes rise from white tubes like flowers in darkness. These animals are giant tube worms, rooted beside fluids that would poison many familiar creatures. An adult Riftia pachyptila has no mouth, no stomach, and no functional digestive tract. Yet it grows rapidly, builds long tubes, repairs tissue, and produces offspring. The central mystery is immediate. How does an animal eat when no opening receives food? Its answer lies inside a hidden organ packed with bacteria and supplied by specialized blood. The worm gathers raw chemicals from two environments without swallowing either one. Oxygen arrives from seawater, while hydrogen sulfide rises from cracks in volcanic rock. Inside the body, symbiotic bacteria combine those ingredients to manufacture organic matter. If you watched the waving plume alone, the animal might seem to filter invisible prey. Direct evidence instead reveals a partnership powered by chemosynthesis. Not ordinary feeding. The vent supplies chemical energy. The plume gathers reactants, and circulation connects them underground. Every movement around the tube follows this fragile intersection between toxic fluid and oxygenated sea. Too far from the vent, fuel disappears. Too close, heat and chemistry become dangerous. The mouthless giant survives by occupying the narrow border where separate chemical worlds meet. Riftia pachyptila inhabits hydrothermal vents in the eastern Pacific Ocean. Adults live inside protective tubes attached to hard surfaces around active fluid flow. Only the red branchial plume extends into seawater, increasing contact with dissolved chemicals. The plume's color comes from abundant hemoglobin within a dense network of blood vessels. That hemoglobin can transport oxygen and sulfide, without the sulfide immediately disabling oxygen carriage. Blood carries both compounds toward an internal organ called the trophosome. Trophosome fills much of the body and contains vast numbers of symbiotic bacteria. Those bacteria oxidize reduced sulfur compounds and use released energy to fix inorganic carbon. The resulting organic compounds support the worm's metabolism, growth, maintenance, and reproduction. This process is chemosynthesis because chemical reactions, rather than sunlight, provide usable energy. The worm does not merely house bacteria. Its anatomy regulates their supply and environment. When sulfide concentrations rise, the plume absorbs more fuel and circulations distributes it internally. When vent flow weakens, delivery falls, and the partnership faces an immediate energy shortage. The worm can also absorb some sulfide across a buried body extension, contacting vent fluids. That extension reaches within the tube toward porous substrate, where reduced chemicals circulate. This arrangement separates sensitive exchange surfaces while drawing compounds from different places. Protective molecules limit sulfide toxicity, but the system still operates within physiological boundaries. High-affinity respiratory pigments help capture oxygen in habitats where mixing creates steep gradients. The animal's closed circulation moves gases and nutrients across a body that cannot browse. Its tube provides support and defense while positioning the plume within productive flow. When disturbance arrives, the plume retracts rapidly into the tube, reducing exposure to damage. After danger passes, it extends again, restoring chemical uptake and therefore bacterial production. This Q-action adaptation sequence turns simple withdrawal into a trade between safety and energy. The partnership begins differently from the fully formed adult condition. Larval giant tube worms disperse through seawater before settling near suitable vent chemistry. They do not inherit a mature trophosome already filled with the required symbionts. Environmental bacteria enter during early development. After settlement initiates transformation. Current evidence indicates symbionts enter through larval skin and migrate into developing tissues. Bacteria colonize developing tissues, and the trophosome expands as the gut regresses. This developmental transition solves one mystery while creating another about partner recognition. The larva must encounter appropriate bacteria within a habitat that may be scattered and short-lived. Chemical cues probably help guide settlement, although the complete decision process remains unresolved. Settling beside active sulfide is beneficial only if temperature and oxygen also remain tolerable. Once attached, the juvenile commits to a fixed position as the vent field changes around it. Rapid growth can raise the plume toward mixing water and secure access before competitors crowd the site. Documented vent communities can develop quickly after eruptions create fresh surfaces and new fluid outlets. Tube worms often become prominent foundation animals, altering water flow and providing physical habitat. Their success, nevertheless, depends on continuing geological activity beneath the colony. The central evidence comes from anatomy, chemical measurements, controlled physiology, and molecular analysis. Anatomy shows the missing adult gut and the bacteria-filled trophosome. Chemical measurements trace sulfide, oxygen, carbon compounds, and metabolic products through the partnership. Physiological experiments reveal transport capabilities, though laboratory conditions simplify turbulent vent mixing. Molecular analysis identifies symbiont functions, but genetic potential does not prove every pathway is active. Together, these methods support chemosynthesis without implying that every metabolic exchange is fully mapped. Scientists still debate how nutrients move from symbionts into host tissues at different life stages. The host may digest some bacterial cells while also receiving compounds released through regulated exchange. Both routes can operate, and their relative importance may change with conditions. Rates measured in collected animals may shift after pressure, temperature, or flow is disturbed. Chemical measurements preserve environmental context, but external readings cannot reveal every internal transaction. The mouthless condition is therefore verified, while many details of cellular trade remain under investigation. If you descend toward the colony, its apparent stillness dissolves into continuous chemical management. Plumes bend with currents, exposing exchange surfaces to alternating pulses of oxygen and sulfide. A favorable pulse increases uptake, circulation supplies bacteria, and new tissue can follow. A harsh surge triggers retraction, protecting the plume but temporarily reducing the energy supply. Repeated choices are physiological rather than conscious, shaped by receptors, muscles, and natural selection. The worms also connect the rocky crust to a larger food web. Predators and scavengers consume plumes, tissues, mucus, or organisms living among the tubes. Organic matter produced without sunlight therefore passes into crabs, fishes, microbes, and surrounding sediments. The tube thickets create shelter and surfaces, increasing structural complexity around active vents. When fluid outlets migrate, colonies may decline while new patches begin elsewhere. This turnover makes dispersal essential, because every successful colony ultimately depends on another suitable vent. Larvae entering currents face vast areas where settlement would fail. When vent chemicals provide the right cue, descent and attachment can convert chance into colonization. That adaptation spreads risk across space. Yet ocean circulation can also carry larvae away from all habitat. Deep-sea mining proposals have increased concern about disturbing vent fields and their biological connections. Not every vent hosts Riftia pachyptila, and communities differ among ocean regions. Protecting representative sites requires knowing which populations exchange larvae and which remain isolated. Recovery cannot be assumed merely because tube worms grow quickly under favorable conditions. A crushed tube, sealed fluid pathway, or altered current may erase the chemical border they require. Their extraordinary nutrition, therefore, reveals dependence rather than independence. Future evidence must track symbiont entry, nutrient transfer, dispersal, and vent change across complete life cycles. High-resolution imaging could follow juvenile development. Without treating rare snapshots as a universal sequence, chemical sensors beside living plumes could connect natural pulses with retraction and growth. Genomic activity measurements could identify exchanges while remaining anchored to measured environmental conditions. Population connections will remain uncertain until larval origins and destinations are directly established. If you return to the white tubes, the red crowns now mark an active boundary. Seawater delivers oxygen from above, and fractured crust releases sulfide from below. Blood gathers both, transports both, and keeps their dangerous meeting under physiological control. Inside the trophosome, bacteria turn geological disequilibrium into compounds an animal can use. What looks like feeding without a mouth is a distributed system spanning rock, water, blood, and cells. Unknown details concern regulation and exchange, not the partnership's fundamental source of energy. No sunlight reaches this colony, yet surface-derived oxygen still links it to the wider ocean. No stomach digests a meal, yet bacterial metabolism continually builds living material. The vent may eventually fail, ending the local supply and forcing the lineage to disperse again. The giant tube worm lives without a mouth or stomach by making an entire chemical frontier its feeding organ.